All right, I think it's 11, so we're going to go ahead and get started. So first of all, good morning. Welcome to everybody that's joining us in person, and also welcome to everybody that's joining us on the webinar. Just a quick housekeeping function. If you are joining on the webinar, there is a Q&A box. And so if you have any questions during the seminar or at the end, you can put them in the Q&A, and I will ask those to Dr. Hopper at the end of the seminar. So it's really a great pleasure for me to introduce today's seminar speaker, Dr. Anita Hopper, who is a professor of molecular genetics at The Ohio State University. Dr. Hopper got her undergraduate degree from the University of Illinois in Chicago, and then she carried out her PhD studies at the University of Illinois, Urbana-Champaign. And this is just a fun fact for this audience, but one of Dr. Hopper's classmates in graduate school was none other than our previous director, Dr. Linda Birnbaum. And the two of them actually got to reconnect yesterday. It had been, 40 years since they last saw each other. Dr. Hopper carried out her postdoctoral studies at the University of Washington, and then she started her independent lab at the University of Massachusetts Med School, where she also received tenure. In 1979, her lab moved to Penn State University, where she stayed until 2006 and rose all the way up to the rank of distinguished professor. In 2006, she moved her lab to Ohio State, where she became chair of the Department of Molecular Genetics. Over the years, Dr. Hopper has won a slew of awards and honors. She is a fellow of the American Academy of Microbiology, the American Association for the Advancement of Science, the American Academy of Arts and Sciences, and in 2021, she was elected as a member of the National Academy of Sciences. Throughout her career, she's also been an incredibly supportive member of the RNA Society, serving on both its board of directors and as its past president. And she's been honored with not one, but 2 Lifetime Achievement Awards from the RNA Society, one for service and one for science. Most of Dr. Hopper's career has been devoted to the study of one particular RNA, and that, of course, is a tRNA. tRNAs are these wonderful little RNAs, which of course carry out the incredibly important role of decoding our genome. And Dr. Hopper's work on tRNAs has really illuminated how tRNAs get processed and how tRNAs get to where they need to go inside the cell. I think one of her biggest discoveries, which is actually incredibly relevant to the mission of this institute, is she discovered a retrograde transport pathway for tRNAs, whereby tRNAs actually accumulate in the nucleus following environmental stress. And I just want to close by saying I had a very selfish reason for inviting Dr. Hopper today, and that is because we have a very shared interest in tRNA splicing. Anytime I give a talk on tRNA splicing, the number one question that I get asked is, what on earth is the intron doing? And there is no better person to answer that question than Dr. Hopper herself. So without further ado, please join me in welcoming Dr. Hopper to the NIEHS.
I want to hide behind the podium. Thank you for that really generous introduction. And I'm delighted to be here. And Robin told me that this is the first seminar since COVID. So that's amazing. So almost 3 years. So can you all hear me if I have this turned this way? Okay. As Robin said, I work on tRNAs, and today I would like to talk about their lives and deaths and the function and splicing of introns from tRNAs. And I am enthralled with the complexity and the regulation of this molecule. So I have to, whoops, we are not beginning. I think I can just go back. Okay. So I just want to be sure everybody's on board here. I shouldn't have to explain this because you have Robin here, but tRNAs are the original small non-coding RNAs. They've been known for 60 or 70 years. And on the left here is the 72, 73 nucleotide tRNA folded in its secondary structure with 4 stems and 3 loops. And then on the right is the 3-dimensional structure of tRNA folded by the matching nucleotide, color matched nucleotides in the 2D structure. And it's absolutely beautifully organized, evolved for its essential function, where an amino acid gets added here at the 3' end, and the anticodon is located here. And so this tRNA can deliver an appropriate amino acid to a codon of a messenger RNA that's being translated on the ribosome. So tRNAs perform this essential function of decoding the genome, serving as a conduit from the genome to the proteome. In addition to this essential function, tRNAs serve a variety of additional functions. For instance, as the Hindenburg lab showed, in response to stress, an important uncharged tRNAs, they can serve as retroviral replication as a primer for RNA synthesis. They have functions in apoptosis, etc. So it should come as no surprise that these tRNAs have very important function in human health. For instance, anything that changes the cellular pools of tRNA, either upregulation or downregulation of individual or the entire pool of tRNAs, results in metabolic and neurological diseases and cancer. Any step in the production of these tRNAs and post-transcriptional modification or processing, I'll be showing those steps, cause a variety of diseases, both mitochondrial and diseases and neurological diseases. And a recent field, just emerging field in tRNA biology, is the production of tRNA fragments. So under stress, tRNAs can be clipped into half-sized molecules here in the anticodon, or quarter-sized molecules into either these 2 loops. And they serve a variety of non-canonical functions in, they can regulate translation, they're involved in epigenetic gene transcription, they can function as microRNAs, and they can promote metastasis. And this field is just emerging. New fragments and new functions of these fragments are being discovered at a regular pace. I should say, if you have a question, please feel free to interrupt. I love to interact with an audience, so please do that. Okay. We work with budding yeast, but almost everything I'm going to tell you, except for one thing, and I'll point it out, happens exactly the same in human cells. So, and I'll show you why we use yeast as a model system as we go along. And this is our working model for the evolved functions and production of tRNA, and it's unexpectedly complex and quite beautiful. So unlike messenger RNAs, and most people are familiar with the biology of messenger RNAs, which so much of the post-transcriptional steps happen at the site of transcription, at the loci of transcription. Yeast tRNAs sample the entire nucleus, the nucleolus, the nucleoplasm, the nuclear rim, and almost the entire cytoplasm in the process of their biogenesis. So like all RNAs, they're made incorrectly. They're too large. They have leader RNAs, leader sequences and trailer sequences at the 5' and 3' termini, and a fraction, a subpool of the tRNAs contain introns, and I'll be spending some time talking about introns. In yeast, it's 22% of the tRNA genes are intron encoding. In rats and humans, it's about 6%. I have scoured the databases for tRNAs, and from archaea to lower eukaryotes to higher eukaryotes, I really can't find an organism that doesn't have a subset of their tRNAs without introns. So introns can serve function of tRNA genes. Okay, so this is the initial transcript. In yeast, this occurs in the nucleolus, and the 5' processing event, removal of the leader, and one particular modification are thought to occur in the nucleolus. And then the tRNA travels throughout the nucleoplasm, where it gains more modifications, and the 5' end is, I said, mature. The 3' purple sequences get removed, and then the 3' CCA, which is essential for amino acylation, gets added. And then this end-matured, partially modified intron-containing tRNA is escorted out to the cytoplasm for its next step. And this next step, actually, this is the only difference I know of between the organisms, is in vertebrate cells, the removal of the yellow sequence, the introns, occurs in the nucleoplasm, but in budding yeast, fission yeast, protozoa, plants, and almost every organism I've been able to inspect, the intron gets removed in the cytoplasm. And in budding yeast and fission yeast, it actually gets removed on the surface of mitochondria. And the surface of mitochondria in many, many organisms serves as a site of post-transcriptional processing events. And it's not quite clear why this organelle has been chosen for RNA processing. And this happens to be the site of the tRNA splicing endonuclease, which I'm sure everybody in this room has heard the beautiful work on the splicing endonuclease heterotetramer from Robin. But in yeast, this enzyme lives on the surface of the cytoplasmic surface of mitochondria, and that's where the yellow sequence gets spliced out. This intron that's removed from the precursor tRNA is rapidly and efficiently destroyed, so it's almost never viewed in eukaryotic cells. And then there's the two halves of the tRNA that get ligated together, more modifications. So about half the modifications, tRNAs are highly modified. So yeast has about 13% of the tRNAs are not A, G, U, or C, and yeast encodes 25 different modifications. There's about 130 different modifications that have been found on tRNAs, so very highly modified. About half of them get added in steps inside the nucleus, and many of them get added after splicing in the cytoplasm. And so finally, after the function of scores of enzyme, we end up with a mature tRNA that can receive an amino acid at its 3' end and participate in protein synthesis. But that is not the end of the story. Unexpectedly, and actually was discovered on 3 continents, almost identically, almost at the same time. My lab, Yoshishi's lab in Japan, and another lab in Cambridge, England. What we learned is that these tRNAs travel back to the nucleus in a process we call tRNA retrograde nuclear import. And if the cells are well fed and happy, then these tRNAs are re-exported to the cytoplasm in a step we call tRNA re-export. And because the tRNAs will accumulate in the nucleus under stress, this serves as one function of this crazy retrograde pathway where the tRNAs are removed from the cytoplasm, the site of protein synthesis under stress. So it's almost a way of downregulating protein synthesis under stress conditions. Okay. So today I'm I'm going to talk about the movement of tRNAs from the nucleus to the cytoplasm. I'm going to talk about some in-process work on the turnover and the likely function of tRNA introns. And I'll have a few words about ongoing work on tRNA retrograde nuclear import. And we were particularly interested in studying this step here, the movement of tRNA from the nucleus to the cytoplasm, because it's absolutely essential for this step to occur, because protein synthesis occurs in the cytoplasm, and you have to deliver the tRNAs, which are encoded in the nucleus, to the cytoplasm. And there was, for a very long time, a single nuclear exporter that conducts this function, and it's conserved throughout evolution. Almost every organism has this gene. It's called exportin T in higher eukaryotes and loss one and yeast. And it is an unessential gene in every organism that has been removed from, including human cells. And so what that says to a person like me, I'm a geneticist, it means that there's other genes that are doing this, because you must move tRNA from the nucleus to the cytoplasm in order for cells to live. And we were highly motivated to find out what else was in this function. And I put the results here, but I'll take you through a few experiments that talk about that. Okay. So I said I'm a geneticist, and I have this model, which is listen to the cells. Used to be, I would kind of guess what gene might function and what process and set up a selection. But nowadays, you don't have to do that. You can unbiasedly look through your entire genome and find out what genes are performing what function that you're interested in. And I'll show you how we do this. And one of the reasons why we use yeast, this was work done by a former graduate student, Jing Wu. So we can take and interrogate really the whole proteome of yeast. It's about 6,300 genes, and ask gene by gene, do you perform a function that's important for tRNA biogenesis? And the reason we can do that is there are collections stored in 96 well plates, about 4,900 strains that have deletions of unessential genes. They're ordered, they're tagged, and they're stored in these 96 well plates. And so you can ask, what do each of these non-essential genes do? Because you can take out a yeast cell lacking that particular function and ask, say, whether it's important for the process you're studying. And then about both in vertebrates and in yeast, about a third of the genes are essential, so you can't remove them. But there's two other collections, and I'll show you that in a minute, in which essential genes have been converted to conditional mutants, temperature-sensitive mutants. And so you can score their function at a non-permissive condition at high temperature. And they're also arrayed on these 96 well plates in order and tagged and identified. So Jing Yang used these collections, which cover about 90% of the proteome. These are all the ORFs. And she was able to grow them in cultures and deep microtiter wells. And then she improved the RNA isolation method, so she could do it in just a matter of a few hours, isolating just the small tRNAs, highly enriched RNAs from the total tRNA pool, from the total RNA pool from these cells. And she developed a non-radioactive northern procedure so she could monitor the processing events. And of course, her work was very dependent on luminaries in the field. So many years earlier, the yeast field developed the whole genome deletion collection. This is done by Phillipson, Mark Johnston, and Ron Davis. headed this large project, and this is what it's telling you about. Every single unessential gene was replaced by a drug marker and then tagged on both ends and then stored in these 96-well plates. And that was done pretty early, 1999. So this has been a valuable resource for use. And then a little bit later, Charlie Boone and Phil Heater isolated temperature-sensitive mutations in the remaining essential genes, and about 80% of the essential genes are stored in these two collections, and they're largely non-overlapping because they took different methods for getting it. So these reagents were available for us to use in our screen. Okay, so this is another drawing of the processing pathway of tRNA biogenesis. And we were very interested in this step, as I told you, of the movement of tRNA from the nucleus to the cytoplasm. And we were trying to look through these yeast collections to see if there was any other gene other than the canonically known loss1 exportin T that would perform the same. So when you remove the intron, the tRNA changes size. The wild type cells, by this northern procedure we used, we can see the initial transcript, we can see the end process intron containing transcript, and we can see the mature tRNA. And a cell that's inhibited for tRNA nuclear export, you accumulate this end process intron containing tRNA because the tRNAs are, here in the nucleus, away from the heterotetrameric tRNA splicing endonuclease here in the cytoplasm. So they, the intron, the pre-tRNA, and the splicing endonuclease don't get together efficiently. And so you accumulate this end process intron containing tRNA. So we were in our screens looking for any mutants of yeast that might have a similar phenotype as this loss1. But along the way, we realized that even though tRNAs have been studied for like 70 years, that there were unknowns in this biosynthesis pathway, including every step I've highlighted in red. And in blue were mutants that we found in processes we were able to unravel due to this screen. I'll just show you this one and intron turnover as examples. Okay, so we were looking for cells that, when we isolated the RNA and did the northern analysis, would accumulate this intron containing tRNA. We actually found 55 novel genes that did this, and I won't go through the rest of most of them, but two of them were here. So this was done by Jing Yang, as I said, who not only established the methods, but went through the entire collection with help of this high school student, Alicia Bao, who then stayed in the lab to do undergraduate research, and then later on joined by two other members of the lab. And what we discovered was surprising to us. We discovered that there were two additional exporters. One was the heterodimer MEX67 or MTR and MTR2, or these are the vertebrate names for these. And this heterodimer was known to perform a function in the movement of mRNAs from the nucleus to the cytoplasm, but had no previously known role in tRNA nuclear export. And the other protein we discovered was CRIM1, which is called exportin T in vertebrate cells. And this was a previously known exporter of proteins from the nucleus to the cytoplasm with no known previous function in tRNA nuclear export. But we're highly confident that they play a role in tRNA nuclear export because, first of all, they accumulate this unspliced tRNAs. That was what we were searching for. So, of course, they did that. We, by RNA fish, could show that the tRNAs were accumulating in the nucleus. by genetics, we could show that these mutations of these genes genetically interact with LOS1. So, for instance, if we overproduce MEX67 MTR2, it totally substitutes for LOS1. We can delete LOS1 from the genome and just have extra of this heterodimer, and the cells don't know that there's been a problem with tRNA nuclear export. And this particular mutation is synthetically lethal with LOS1. So that means they have a shared function. So we knew they were on the same pathway by these genetic studies. But even for me as a geneticist, the most convincing evidence is that the proteins that we identified assemble in vivo into nuclear export complexes with intron-containing tRNAs. So I'll just show you one example of that. And so we know we have 3 tRNA nuclear exporters. And the question was, do they really perform exactly the same function? And do they have exactly the same fidelity? And I'll spend a little bit of time telling you what we learned and found to be surprising. Okay. So a little bit about nuclear exporters in general. So there's a family of smart proteins called important beta family. In vertebrate cells, there's 22 members. In yeast, there's 14 members of this pathway, and they're RAN, which is a small nuclear GTPase binding proteins. And this members of these families either are involved in movement of macromolecules from the nucleus to the cytoplasm or from the cytoplasm to the nucleus. And the way they function in nuclear export is the exporter can bind the cargo, and we're looking at tRNAs for this experiment, but controls are messenger RNA or other proteins. In the presence of RAN in its active GTP bond form, and these smart proteins find their way across the nuclear pore, and then in the cytoplasm, there's a RAN gap, which helps the hydrolysis of RAN GTP to RAN GDP. There's a confirmation switch of the exporter. It releases the cargo from the export complex. So this is a work plan for these particular members of the family. LOS1, exportin T is one of them. CRIM1 is another member of this family. And so we start with tag proteins, and everything we do is endogenous quantities. So we replace the normal protein with a functional tag version that's expressed under the normal promoter. And we try to trap this complex, and we do it in 2 ways. One way we do it is we translinkly express a non-hydrolyzable RAN GDP. And the other way we do it is we use formaldehyde cross-linking. So we try to trap this normally transient complex in the cells, and then co-IP, and then analyze which proteins are associated with that complex and which RNAs are in that. MEX67, the mRNA nuclear exporter, is not a member of this family, so we skip this particular step, the trapping of the non-hydrolyzable RAN GTP. Okay, I'm going to skip this particular, the left side, which does the studies for LOS1 and 67. I'll just show you these newly published ones for CRIM1. So this is a protein stain of the proteins we purify by the pull downs. They're pretty good, 1-step purification. And this is a Western, because the proteins are also tagged with GFP. And so we can see that in this particular experiment, these proteins are CRIM1 tagged with GFP. And we also can pull down RAN protein. And then we isolate the RNA, reverse the cross-links, isolate the RNA, and then see what's in it using RT-PCR. So in this case, we have a primer to the intron of the tRNA and another primer to the 5' end. And you can see that we can amplify this tRNA, precursor tRNA, in cells that have RAN locked in the GTP bound form, but not in the inactive GDP bound form. And we don't see anything in the wild type cells that don't have the tag protein. So that says that this CRIM1 can bind an intron containing pre-tRNA locked in the nucleus. And so now we can track this tRNA nuclear export complex. We have a lot of controls that I think demonstrate that this is in fact what we're doing. First of all, positive control, another substrate that was known for this CRIM1. And then down here is we amplify mitochondrial tRNAs. So this is a separate compartment of cells. And we have to really do a lot of amplification, I think 37 cycles of our PCR. And it just serves to see how much non-specific RNA we pull down in these. And you can see in every single lane, the amount of non-specific RNAs is the same. So that says to us that the specific pull downs that we see is a real result. So doing this kind of work for all three now, exportin T, the mRNA nuclear exporter, and now the tRNA nuclear exporter, we conclude that there are at least 3 parallel pathways that move tRNAs from the nucleus to the cytoplasm. And then the question is why? Why would you have 3 exporters, and do they have the same function? And so we repeated this same experiment, which was done with a single tRNA. And in yeast, there's 10 families of tRNAs that have introns. So we looked at the other 9 and try to see how well those tRNAs made nuclear export complexes with each of the 3 pathways we have found. So that's the workflow. And this was the results. And in each little box here, we compare the amount of a particular tRNA in a particular tag strain with the amount that co-purifies in these nuclear export pull-down experiments. I don't expect you to see the data, but we plot it as the relative efficiency of pull-downs. And we see that some tRNAs are like this tRNA ILU are pulled down efficiently by all 3 different pathways, whereas other tRNAs are pulled down by one exporter, not so efficiently, but pretty efficiently by others. So these nuclear exporters have substrate specificity or substrate preferences, which was a surprise for us because, as I showed you in the first slide, all these tRNAs look pretty much the same in their 3-dimension. And we were thinking that they should all make complexes the same way, but they do not. And that's been a surprise for us. So, mini summary. We see that endogenously expressed yeast proteins can assemble into in vivo in nuclear export complexes with intron-containing tRNAs. And that tells us that there are at least 3 parallel acting tRNA nuclear exporters, but the 3 pathways have individual preferences. And we have some experiments that show they behave differently under different stress conditions and different environmental stress conditions. And this could be powerful because it could mean that you can adjust the pools of tRNA that are in the cytoplasm by differential use of one or more of these export pathways. And so we're working on that to see if tRNA nuclear export is a new level of regulation of gene expression under stress conditions. Okay. And then we also want to know if each of these pathways has the same fidelity. So it's really, really, really important that tRNAs are 100% accurate and form completely correctly, because they're used iteratively in protein synthesis. And if they're not functioning appropriately, then you start making aberrant proteins, which will go back and have faults in replication, et cetera, and you will build up over time populations which are not functioning correctly. So there's all kinds of levels of quality control that that play a role in the fidelity of tRNA at many steps. But we found that there are actually errors in tRNA nuclear export. So this was work by a former postdoc, Emily Kramer, whose picture will show up here shortly. And what she found were two different types of errors. So one kind of error, we found that sometimes the tRNAs exit the nucleus before the ends are clipped off, and then they get spliced in the cytoplasm, and then we end up with tRNAs that can't really function in protein synthesis because they can't receive an amino acid at the 3' end. And the other kind of error we found was that some tRNAs exit the nucleus before they receive all their modifications that occur in the nucleus, and they go out into the cytoplasm. And some of these modifications cause misfolding of the tRNAs and decreased stability, but also errors in decoding the genome. So we were kind of surprised to see this, but it makes some sense because there's all these competing processes occurring in the nucleus. There's scores of post-transcriptional processing and modification that's competing with the nuclear export process. And so you can imagine that sometime tRNAs get out of the nucleus prematurely before everything has happened. And so we were interested in studying this further. And what Emily Kramer found was this is a second role for the nuclear import process, because these error-ridden tRNAs increase remarkably if we inhibit the import of these tRNAs back to the nucleus. So this nuclear import re-export process serves a role in nuclear tRNA quality control. So now that we have the 3 exporters, we want to know which of them are doing these errors, which of them are error prone. And this is some beautiful work from Alyssa Conti's lab, where she obtained a co-crystal of the exportin T, it's a RAN binding protein, in the presence of RAN GTP and tRNA. And you probably can't see, but it's very, very cozy here. It will not accommodate leader containing or trailer containing tRNAs. There's just no room in this structure for extra sequences at the 5' or 3' end. So what that means is that this particular exporter, when it's functioning, ensures that there's end processing before nuclear export. So how about the 2 new pathways that we discovered? And so the question is, do the MEX67, the mRNA export pathway, CRIM1, do they have the same function in monitoring the 5' end of the tRNA before moving it from the nucleus to the cytoplasm? And the way we do these experiments is we again use the very similar RT-PCR function, and we look to see whether there's complexes of this tRNA that has the intron containing, a leader containing tRNA in the cytoplasm. And what we can see is that the MEC67 can bind these tRNAs before removal of the, so now we're looking at the leader, not the exon, can bind leader containing, intron containing tRNAs, and presumably take them to the cytoplasm where they get spliced. But we didn't see any accumulation of these with loss1, the exportin T, which we knew from the crystal structure should happen, nor from the other pathway we discovered CRIM1. And even if we overproduce these exporters, so if we overexpress like loss1, exportin T, we still do not see any binding of it with the end extended tRNAs. But MEC67 is very efficiently binds these pre-tRNAs. So it can export tRNAs prior to end maturation and deliver them to the cytoplasm. That's bad news for yeast, and they shouldn't be doing that. So there's low fidelity of the mRNA nuclear exporter for moving tRNAs prior to end processing. And so what happens to those? So we looked to see whether they're destroyed, and we looked at a couple of different pathways. We looked at 5' to 3' decay in the cytoplasm and in the nucleus, and 3' to 5' decay in the nucleus and the cytoplasm. And what we learned is that these leader-containing tRNAs get moved back to the nucleus and get destroyed by the 3' to 5' exosome in the nucleus. So this retrograde pathway is playing a role of collecting the tRNAs that are inappropriately delivered to the cytoplasm, destroying them before they end up mucking up protein synthesis. And this is work done by former postdoc Canal Chatterjee and another undergraduate, Will Marshall. So this is a cartoon that the tRNAs can get out of the, this is Will's cartoon, get out of the nucleus to the cytoplasm. And we haven't tested whether they have 3' end, but we think they probably do, because 5' ends get processed before 3' ends. And the tRNAs are then, they get spliced in the cytoplasm, they get retrieved back to the nucleus, and then destroyed in this 3' to 5' direction, and not in the 5' to 3' direction, and probably because they're initial transcripts and they have a triphosphate on the end, which the 5' to 3' nucleases need a monophosphate. And also there's a report from that the 5' ends can be capped. And so those would also be inhibited from being destroyed in the 5' to 3' direction. What we don't know is whether they also can be destroyed in the cytoplasm. And my favorite idea is that when they return to the nucleus, maybe they're given a second chance. Maybe they can get their ends processed and then come back out whole. So you don't really have to destroy the whole tRNA, you can repair it. And so I have some students working on that process. Okay, so I'm going to summarize here. Well, how much time?
About 15 minutes.
15 minutes, okay. I'm going to summarize here. So we have 3 pathways functioning in parallel, and they seem to have different fidelity and different substrate specificity, and that was unanticipated for us. And I think I'm going to skip this so I have more time to talk about the destruction, turnover of the freed intron, and what we think might be possible functions. So cells make an enormous number of tRNAs, intron-containing tRNAs per cell cycle. It's about 600 molecules of 600,000, molecules of free introns are generated every cell cycle, and you almost never see them. So they are very efficiently turned over, and we were very interested in learning something about that process. A little bit about introns. They're very different tRNA introns from messenger introns. First of all, they're always located at the same place in the pre-tRNA, one nucleotide downstream of the anticodon. The splice junctions have no consensus sequences. The mechanism of catalysis, as you know from Robin, is a tetrameric protein. It's not an RNA catalyzed reaction. And the intron-containing tRNAs can not function in protein synthesis. There's no alternative function, alternative splicing for these molecules. And as I said, they're rapidly turned over. Yet, possession of introns is conserved from archaea to humans. So the question is, why? Why preserve this structure inside of tRNAs if there's no possibility of alternative use of them, and the tRNAs cannot function in their normal canonical function if they possess it? Okay. And furthermore, as I told you in the beginning, every organism I've looked at has at least one family of tRNAs where all reiterated copies of it have an intron. That means you can't decode the genome unless you have the ability to splice tRNAs. Okay, so this is the same genome-wide screen I showed you before, but in this case, we were on a hunt for mutants that accumulated the introns, so virtually is gone in every other cell, but we did find a mutant that accumulated free introns. So we knew that we had discovered something about the turnover pathway. And this is just the data showing that, in fact, this mutant does accumulate the free intron, and that if we provide the wild type protein, the intron no longer accumulates. And further studies of this generated this particular pathway where the tRNAs come out of the nucleus to the cytoplasm, and then they're spliced, and then they're ligated together. But this ligase has a kinase activity, and it turns out that that kinase activity can phosphorylate the 5' end of the free tRNA, and then it becomes a substrate for this 5' to 3' exonuclease, and it is degraded. And I thought, great, now we understand how tRNA introns are turned over. We're really in good shape. But the same was high school student, now undergraduate, and there's 10 families of these introns, and I thought a good set of experiments for this undergraduate would be, look at the other 9 families. I'm sure they're all going to function the same, but let's just take a look. And I was wrong again. So this is the tRNA that we had done the genome-wide screen on. This is a mutant lacking the exonuclease and a mutant lacking the kinase activity of the ligase, and both accumulate the free intron. So that's where our starting point is. But then as we did these experiments, we found that there are at least 5, perhaps as many as 8 separate pathways for turning over these introns. So for here, we have an example of 2 members of the 10 families that are dependent on that same 5' to 3' exonuclease, but they're not dependent on the kinase activity of the ligase. That means we're missing our RNA kinase, and we haven't found it yet. And then we have sort of the yin yang of this. There are 3 members of this family that are dependent, whose intron turnover is dependent on the kinase activity or ligase, but it's independent of the 5' to 3' exonuclease and every other exonuclease we've tried. So we're missing an unknown nuclease that would function in collaboration with this kinase activity. And then we found 3 families of the tRNA that are independent of every enzyme we've looked at, and we've looked at every annotated RNase and every annotated kinase in the yeast genome, and we have not found this. And then one of these tRNAs, only one of the 10, makes circles, and so you need an endonuclease to destroy that particular member of the family. And so Alicia, and joined by Sarah and another Andrea Katie, have been looking to see if we can identify the rest of those. We're now headed in another genome-wide screen to look at these other 10 families because we're not good guessers. We're just can't find what we're looking for in the annotated genome. But it raised a question to us: is why would you have 5, at least, maybe 8 different pathways for turning over these linear RNAs that look pretty much the same and are released from this precursor tRNA, and start thinking, well, maybe these tRNAs are serving a function and there's regulated turnover of the introns. And that became even more apparent to us when we started looking at environmental conditions that may affect the tRNA turnover. And we were very surprised to see that there was stress-specific tRNA species, families, specific tRNA intron turnover pathways. So start here. Let's see if I can. All right. Oh. So start up here. So this is one of the tRNAs, tRNA trip. We treat these cells with H2O2 as fast as we can treat them. The cells stop transcribing the tRNAs, and they also start exporting them from the nucleus of cytoplasm. And this intron, which is really pretty much inapparent in cells grown in normal media, starts accumulating. And even though we're not making any more tRNAs over this 2-hour period, this spliced intron is just stable as a rock for hours. Whereas, and that's this red line here. Whereas other members, like this orange line, are semi-stable, and other ones, like this one, there's a little accumulation for a few minutes, and then it disappears. So this means that not only are there the different pathways for turnover, but they're regulated by different environmental conditions. And so again, that's why is there's some stress-specific function of these introns that they have to be translated under, that they have to be regulated under particular conditions. Okay, I'm going to skip that one. And most amazingly, both Alicia and Gina Nostromo, who is currently working on this, did a little bit of bioinformatics, and we shockingly learned that these introns have long, uninterrupted sequence complementarity to messenger RNAs. So this is the tRNA intron for tRNA ILU. It's 60 nucleotides long, and it goes from left to right. And going down this way are 32 messenger RNAs that have long stretches of sequence complementarity to the tRNA ILU intron. Green is perfect complementarity, blue is GU base pairing, and red is a single base pair mismatch. And some of these are, let's see if I can get it up. Oh, go. Some of them are pretty remarkable. So we've done some additional experiments I hope to show you on this one here. 16 base pairs of 16 bases, perfect complementarity with this particular ORF. And remarkably, for this particular gene, there's 2 stretches of long complementarity. And this actually turns out to be the gene that puts the ILU amino acid on this ILU tRNA. So seems to be some sort of complementarity. And then I'm just showing you one, but for all 10 families, we have complementarity to different tRNAs. And I have this undergraduate who loves calculations, who's calculated based on the sequence of the yeast genome and the ORFs and the probabilities of what the probability would be of having 10 different introns, each with 14 base pairs, minimal stretches of complementarity, and it's impossible. This shouldn't be happening by accident. So it says maybe these introns are doing something. Okay. So our working hypothesis is maybe they're acting like microRNAs and base pairing with particular messenger RNAs and regulating the turnover of those messenger RNAs, or maybe even translation like microRNAs do. Okay. And so we've done 2 types of experiments. I'm running out of time. I want to show you the data. We cut out the introns in all copies of the tRNA gene and then determine what the function is of those, what the levels of the RNAs are that bear complementarity to those tRNAs. So this is all those 32 tRNAs. The one that had 16 base pairs is this guy right here. And you can see we have increased levels of those messenger RNAs compared to 32 randomly chosen RNAs that bear no complementarity that we know about. We don't know the rules yet. So it could be far less than 16 or 14 base pairs that are required. Okay, so we think this means that the complementarity serves an RNA of, turnover. So if when we remove the introns, the messages with complementarity go up, if we were to overexpress these free introns, maybe the messages would go down, and that I think would be strong evidence that they're functioning in the turnover of these messages. And we were kind of stuck there for quite a while because we wanted to generate 3 introns in their normal state with their normal ends. And we wanted them in the right compartment so the tRNAs are free in cells in the cytoplasm. And if you transcribe new ones, they're in the nucleus. We tried a lot of different variations on this, but finally we settled on expressing them from a T7 polymerase. And we can now overexpress these introns that have complementarity to particular messenger RNAs. I'll just show you one last experiment. So we can get varying levels of these free introns with the T7 polymerase, and we can show that when we overexpress the tRNAs, the introns, the more intron we express, the greater the turnover of the introns that bear complementarity, and we don't see any of that turnover with messenger RNAs that don't have the complementarity. Okay. So this comes to our final idea, working model, is that tRNA introns may be evolved and maintained because they can serve as a new category of small non-coding RNAs. and the lines of evidence is conservation throughout evolution, family-specific turnover pathways, stress-specific family-specific turnover pathways, long stretches of complementarity to particular mRNAs, and then removing them or overexpressing them inversely regulate the levels of the mature tRNAs. And so our idea is that these free introns may serve in regulating mRNA levels in response to stress. And I'm pretty optimistic that this is correct because the evidence we're getting continues to support this idea. Okay. I know I'm over, but I absolutely have to say thank you. Everybody in the lab now is up here on the top, and the people who participated in the work I showed are in red. Anybody who has an asterisk after their name is either an undergraduate or a high school student. And I want to give a special shout out for Gina, who's a postdoc in the lab pretty soon on the job market. And she has done just amazing work on what we believe to be the evidence that shows that tRNAs can function as microRNAs. Our work is funded by the NIH and the California at Ohio State. We have a wonderful Center for RNA Biology, and all the undergraduates have been supported by funds from Ohio State that's really dedicated to allowing students to have an undergraduate research program. So thank you, and I'm sorry I went a little bit over. Happy to address any questions.
All right, I think we have time for a few questions. I'm going to start with some from the webinar. And if anybody in the room wants to ask a question, you just need to go up to the podium so that we can use the microphone. All right, so the first question I see in the webinar is from Dimitri Gordine, and that is it known what happens with tRNA export in yeast arrested at G1 or G2?
Say that again.
Is it known what happens to tRNA export at different stages of the cell cycle, like G1 or G2?
There's, as far as I know, no evidence for cell cycle regulation, tRNA nuclear export. Although there's one study that seems to show that it's in mitosis, the tRNAs seem to be preferentially located to the nuclear pores. But that's the only evidence I know of.
Okay.
Hello, thank you so much. It is a fascinating talk. We are working in oxidative stress, and one of the thoughts that came to me is that CRM is also recognizes certain proteins that turn on the oxidative stress response. And one of them is YAP1, which is transcriptional activator of a lot of genes, which protects cells from oxidative stress. So my thought was, have you considered that when, let's say, YAP1 got oxidated, CRM became more free, and so it can change the dynamics of import or export of the tRNAs, and also maybe it can kind of recognize different subtypes of the tRNAs differently. Thank you.
So I'm not sure I heard it all because I have a little hearing deficit and I do a lot of lip reading, but I think you're asking if you can change the dynamics, if that would have a consequence on a pool of tRNAs.
Yes.
And I think that is very possible. There is some evidence for that in human cells that under actually H2O2 stress, there's accumulation of tRNAs in the nucleoplasm, but not every type of tRNA is equally accumulated in the glucoplasm. We haven't done the studies yet, but I'd be shocked if we're not changing the pools available for protein synthesis based on which of these pathways we're using and how they're responding to stress.
Just one clarification. So I was thinking about also very specific protein called YAP1, which is taking out of nucleus by CRM, and it is turning on the antioxidative stress in the cells. So if you can consider that there could be very fine interplay between the YAP1 between CRM working with the YAP and CRM working with the tRNAs.
And then there's this Esteban, I think his last name, he's actually shown that
You can actually adjust the levels of modification of the tRNAs, and they respond to stress, and they actually regulate the turnover pathways of different tRNAs, and under particular stress, you can accumulate high levels of some tRNA and lower levels of others, and those seem to be co-evolved with metastasis genes that use those particular introns. So I think there's a lot of unappreciated levels for regulating the proteome in response to this nuclear export and
Thank you very much.
crisis. Yeah.
I think we have time for one more question. So I apologize to everybody in the webinar. But the last question, and I think we have time for, is do you expect the mechanisms regulating tRNAs are common in all cells? For example, what you showed with hypoxia in yeast, do you imagine that that translates to humans, or are there different pathways?
The same studies have been done by the Fasati lab in human cells, and they respond identically to H2O2. Esteban, his last name I can't remember right now, also shown the same. There's studies in human cancer cells that show that the mRNA nuclear exporter is also functioning in a parallel pathway to the exportin T. There's no evidence yet that the CRIM1 functions in a third export pathway. Actually, in Xenopus, it looks like it doesn't. And I think these same experiments need to be done in higher cells to see just how much of this can be recapitulated in vertebrate cells. But every step that's been looked at, discovered in yeast, turns out to be used in vertebrate cells.
Right, so
And human cells.
unfortunately, we're out of time, but please join me in thanking Anita for a really lovely seminar.